Welcome, friends. Welcome back again. Today we are going to discuss about Mendelian genetics. Yes, the very founding principle of genetics. And he is Gregor John Mendel. Yes, father of genetics. In fact, if we see, he is the person who modernized biology by his experiments and the way he used statistics and mathematics in the subject. So let's begin. Mendel was an Augustine monk in a monastery in Austria, which is now in Czech Republic. He was well trained in maths, statistics, probability, physics, and he was interested in doing something which most of the people at that time was doing, which is plant breeding. Along with his teaching assignments, he also has to look after vegetables which monks eat. And he set up an experiment. Wonderful, isn't it? I don't know what happened, how good was the food to the monks, but definitely they were eating one of the finest experiments results in the history, the pea plant experiment. Yes, this environment is cool, short season of mountains, and they were very good for growing pea. And so he find some of these characters to study the basic principle of genetics, like how genes transform, goes from one generation to other, express or mask each other. So for example, he took some varieties like round seeds and crossed with wrinkled seeds. He used yellow cotyledons and with the green cotyledons, a gray coat or a white coat, a full or a constricted green or yellow pods, or maybe the stem length, like whether it is a tall or a dwarf plant. So one of the thing is, which we already saw, that it's a good environment in the monastery to grow these plants. The and as he was the in charge of vegetables, so he could do some of these experiments. Now this is often asked that whether the experiments set by Mendel were intentional, were they scientific experiment, or just a coincidence. So let's see about the Pisum sativum, the pea plant. Now if you see the pea plant, it's an obligate self-pollinator. What do you mean by an obligate self-pollinator? An obligate self-pollinator is something like this, that this, if this is the plant, okay, this is the flower of the plant. So the reproductive organs of this plant, like the anthers and this ovary, the stamens, now all these are closed by another sepal. Yes. So what happens that the anther from this flower will not go out, or anther from another plant will not come in. Now what Mendel did is he removed these sepals, kept his anthers, pollens on this stigma of the plant, and then he set up the experiments. What does that mean? This means that this was intentional process. There will be no cross contamination because of just by wind or by the insect, which provides a side by side genetic experiment barrier so that flowers don't cross talk, right? Or cross fertilize. Cross pollination required an intentional process. So if he set up an experiment, tall plant crossed with dwarf plant, that was an intentional process. Then he documented this experiment and statistically analyzed it. But at that time, the biologists were unable to understand mathematics, right? And that's why this path breaking experiment was lost in the journal for 50 years, even 35 years of Mendel's death, and then rediscovered by three scientists: Shanmark, Deveris. Yes, Deveris, I know that you know this guy. He did many biological experiments which we study in school textbooks. And Correns. So Hugo Deveris, Correns, and Shanmark rediscovered it independently, and then they realized there are some wonderful thing hidden and lost in this system. One of the reason is that the biologists at that time were unable to understand maths, but also that was the time when Darwin gives his theory on origin of life, and half of the scientific community was favoring Darwin and half of them were anti, but uh, to prove Darwin wrong, but nobody bothered about Mendel's work and it remained lost in the journal for 35 years. Another reason is that this journal was a monastery journal, and that's why it doesn't have a scientific reach at that time. Let's see what he actually did. So he set up an experiment where the one plant, like for example the smooth seeds, is crossed with wrinkled seeds, and then he saw that what the plant in the next generation produce, and he found that this plant produced smooth seeds, which means, so he implies that there are paired factors. Yes, today it is simpler for us to understand. We know that we are diploid organism. There are two chromosomes which has one allele each, and so which segregate into one gamete. Similarly, the other plant, which is this, has two alleles which segregate into this gamete. Now if this thing, the alleles are same, it is called as homozygous. But if alleles are different, like in F1 plant, capital S and small s, it is called as heterozygous. So if we see this one, what is it? Yes, it is a homozygous. Now if we see heterozygous again segregate into two of these alleles like small s and capital S, and if we cross both of these F1 generation, then what we get is, if we put up these gametes in a box like this, capital S and small s, capital S and small s, we get capital S, capital S, capital S, small s, capital S small s and small s small s. So what he said that the capital S is dominant over small s. Why? Because in this heterozygous plant, this follows a pattern, a phenotype. The appearance of this plant, which is called as phenotype, is same as capital S, capital S. So the capital S has an ability to mask small s, right? And so this heterozygous is also has a phenotype of smooth seed, right? Okay, so we understood what is homozygous, what is heterozygous. This SS is the genotype of this plant. The small s is the genotype of this plant, and capital S small s is the genotype of this plant, right? Now capital S has a phenotype of smooth seed. Capital S small s also has a phenotype of smooth seed, whereas small s has a phenotype of wrinkled seed. So there is another term which I used which is called as alleles. Now this is going to be the most important term we are going to use for next 5-6 lectures, because this is the founding principle which tells us that how the gametes segregate and how this phenotype is generated. So what is this allele? Allele is an alternative form of gene. Okay, like here capital S and small s. But this alternative form is present on the same location. So if we talk about a chromosome, if this is the location of S, same location would be for the small s, which means suppose there is an enzyme called as glucose fructose kinase. Now this glucose fructose kinase enzyme will be present on the same location for all the human beings. Suppose if it is present on X chromosome in the P region, for example band number 13, so this location for glucose fructokinase would be same for all the human beings, and that is our understanding of alleles. We'll understand it better as we progress in this course. Okay, so then he set up another experiment in which he crossed the F1 generation, these heterozygous, and what he found that now this heterozygous has a paired factor, so one of the S segregates with the other S, and now we have this combination. And then what he find is that there is a capital S which is smooth seed, a heterozygous also smooth seed, and a homozygous recessive plant. This how he get, if he cross smooth seed, 1/4 of them was wrinkled, and 2 of them were heterozygous and smooth. One of them is homozygous and smooth. So if we see the genotype of this is a unique genotype, these two are the same genotype, and this is another unique genotype. So we have 3 of them as smooth, one is wrinkled, and one of them is homozygous smooth, 2 is heterozygous smooth, and 1 is homozygous wrinkled. This is called as phenotypic ratio and this is called as genotypic ratio. Okay. Now, how do we do that? For this, we use a statistic method, which is the Punnett Square. So for calculating any of this problem, what you need to do is make a squares like this, and then you can put up the gametes like gamete 1 and gamete 2 from one parent, and from parent 2 gamete 1 and gamete 2, and here what you get is the combination of whatever you are doing, like G1 and G1, or here in this case you get G1 and G2, and in this case you get G1 and G2 like we did just now, and then G2 and G2. So basically, if we make this Punnett square, we can simplify this monohybrid cross and we can find out what is the genotypic and phenotypic frequencies. Alright, so there is a problem for you. So if there is a tall plant, okay, where tallness is dominant over the shortness, the dwarf plant, small t. If the homozygous tall plant is crossed with the short plant, what is the genotypic makeup of the offspring and the phenotypic makeup? Pause this video, try it, and then resume, and we both do it together. Okay, so here first we need to find out the alleles, right? What are the alleles which we have? So if it is a genotype homozygous, it is capital T, capital T. The another parent is small t, small t. How many types of alleles are there? There is only one type of allele, capital T, capital T. Here is only one type of allele, small t, small t. So what we are going to do is we will put one of the gamete here, another gamete here from the other parent. See the parent 2 and this is parent 1. And what we are going to get? We are going to get all these are same thing. So basically when we cross capital T with small t, there is only one type of gamete which is formed, which is capital T and small t, and this is a heterozygous. Now let's see what if this heterozygous is crossed with heterozygous. Okay, so what we are, how many types of gamete will be here now? Capital T and small t, because it's a heterozygous. Same way, if we are going to cross this, what we are going to get is capital T and small t. So if we put up this in Punnett square like this, then what we are going to get is this capital T, small t, capital T, small t. So this is from say parent 1 and this one is from parent 2. Now let's see what we get. Capital T, capital T. So this is a homozygous tall. This is capital T, small t. This is heterozygous tall. This is capital T from here, small t from here, and we get a capital T, small t, which is heterozygous tall. And this is our small t from parent 1 or from the parent 2. What we get is finally small t, small t. So in this case, the genotypes, how many genotypes are there? There are 3 genotypes. One is capital T, capital T. The other genotype is capital T, small t. And then we have a genotype which is small t, small t. So the genotype ratio is 1 is to 2 is to 1. Whereas phenotype, if we see, all these heterozygous or homozygous, both of them will be tall and only this one would be short, and so the ratio would be 3 is to 1. So in monohybrid cross, we always get this ratio of 1 is to 2 is to 1, and in phenotypic ratio we get 3 is to 1. How we are getting this? I'm sure you have understood this, and we will be doing more and more number of questions in this series, and then you will understand it better. Like this one. Okay, so now this you have to do it yourself. Okay, pause your video and just tell me what would be the result if a homozygous round plant is crossed with wrinkled seed plant. Homozygous round seed plant with wrinkled seed plant, or the heterozygous round seed plant is crossed with the wrinkled seed plants. Here it is given that the round seed is dominant. It's dominant over wrinkled seeds. So capital R is dominant over small r. So what would be the answer? Pause this video and resume when you are done. Give it a try. And then we are doing it together. Okay, so let's see. What is it? So if there is a capital R and capital R is crossed with small r and small r, then how many types of gametes we have? Capital R. And how many types of gamete from here? Small r. Okay, both of them come together, we get capital R and small r. And this is, so what would be the phenotype of this plant? All of them would be round seeds. All are round. Now let's see this one. Okay, so if it is a heterozygous, capital R is crossed with small r. How many types of gamete this can form? Two types, capital R and small r. And how many? Sorry. Okay, and how many types of this is small r? So let's make a Punnett square in which we have small r and we have capital R and small r. So what would be the product? Capital R, small r, small r, small r. This would be round. This will be wrinkled. Right? What would be the ratio? 1 is to 1. So one of them would be round, one of them would be wrinkled. Now this experiment is also called as test cross. Test cross means that when the plant is round seed plant, you don't know whether it is a homozygous or a heterozygous. To determine whether it is homozygous or heterozygous, what we'll do is we'll cross it with the recessive homozygous parent. And if we get all of them round, we'll say the plant was homozygous. If we get a 1 is to 1 ratio, we will call it as heterozygous. And that's why it is called as test cross. Since it is crossed with recessive parent, homozygous parent, this is also called as back cross. Okay. So these are the two laws which is given by the Mendel. One is law of segregation, which means that there would be two factors. These two are paired, but they will segregate. You know what is this cytological process called? Yes, it is meiosis. basically what Mendel was trying to present is there is a cytological process which is meiosis, in which two gametes segregate. This will understand and will understand more depth when there would be two genes involved, right? And then we have this concept of law of independent assortment would be better, which means if there are two genes like A and B, A has no influence on B and B has no influence on A. Both of them are independent. So A will not influence the phenotype of B, B will not influence the phenotype of A. We are going to see this definitely in the next lecture, which is dihybrid cross, which means two genes together, two genes. Okay, I hope you love this video. Please like, share and subscribe to my channel. Bye bye, see you soon.